Great. So my name is Alexey Arkov. I'm from biology, Department of Biological Sciences at Murray State University. And it's my great pleasure today to host our Murray State seminar as a part of Kentucky in-Breer virtual seminars. So today we're going to hear from Dr. Christopher Lennon from Department of Biological Sciences at Murray State, and also Dr. Gage Jordan from Psychology, Department of Psychology. And Dr. Lennon is going to start first. Go ahead.
Okay. Share my slides. And there you go. Okay, so welcome and thank you for this opportunity to share my work on intervening proteins as environmental sensors. Again, my name is Chris Lennon in the biology department. Okay, so inteins or intervening proteins are mobile genetic elements that invade other protein coding genes. And unlike something like an intron that is removed at the RNA level, inteins are actually translated as proteins within other proteins that then undergo this autocatalytic protein splicing reaction, whereby the two peptide bonds that are holding the intine in place are rearranged into a single peptide bond, and the intine is liberated. And we call this process protein splicing. So why are we interested in inteins? So this ability to rearrange peptide bonds is really useful in protein engineering and has led to the invention of dozens of really exciting biotechnologies. We're also interested in inteins as novel antimicrobial targets, as they're absent in humans and present in some pretty nasty human pathogens. We think this can be a novel avenue to kill these organisms. And then finally, we're interested in inteins for their emerging role as post-translational regulatory elements. And the basic idea is that prior to the protein splicing reaction, the intine-containing protein is off effectively, and then after its activity is restored. And that's what we're going to focus on today. So I want to introduce a term, exaptation, right? And so this is basically evolutionary retooling. And the classic example of this is feathers in dinosaurs, right? So feathers didn't evolve for flight initially, but that's their purpose now, right? Initially, they came to be probably to keep dinosaurs warm, right? And so mobile genetic elements are traditionally seen as junk DNA sequences, molecular parasites that provide no benefit to their host. But it turns out that mobile genetic elements, have been accepted and have actually led to really stunning evolutionary adaptations. Most notably, or one example is a group 2 intron, which has actually led to spliceosomal introns in us, right, which are quite important. Okay, so where are inteins found? Well, it turns out that in the two microbial domains of life, the two prokaryotic domains of life, inteins are abundant. They're found in about 1/4 of all bacteria and about 1/2 of all archaea. And in both these domains, more often than not, they're found within these RRR proteins. So these proteins involved in DNA replication, recombination, and repair. Now, what's really remarkable about their clustering to these genes is that oftentimes, while these are functionally homologous, meaning that they do the same thing in these different organisms, they evolved independently to do so, right? So they are non-orthogonal. Okay. And so how might an intine provide regulatory benefit to a host? Well, one could imagine that your intine-containing protein is translated and it's in this state where it can't splice, and some sort of signal comes along and activates splicing. This could provide regulatory benefit. One could also imagine that splicing under normal conditions will occur rapidly, but then under some sort of stress, any new protein made will be, splicing will be temporarily blocked until that stress is relieved and splicing can be restored, okay? And so during my postdoc and then extending to my time in Murray State, we found examples where both of these types of regulatory mechanisms happen, right? In one example, we see that DNA damage actually induces splicing of the major DNA repair protein, which makes a lot of sense. You have your protein in this inactive state, the stress comes along, and it causes splicing. We also have an example where DNA replication is reversibly paused by, splicing is paused by a number of relevant environmental signals, right? And this is work that started in my postdoc and I've brought here to Murray State. Okay. So we also know that there's other examples of conditional protein splicing where this splicing reaction is tied to environmental conditions. And it turns out that through directed evolution, scientists have even evolved an intein to splice in response to a signal, right? Okay, so the big question here, though, we know that we see this really interesting distribution and clustering in certain types of genes. And we know that when we look at these things, in vitro and in non-native systems, we can see really interesting triggers or things that block splicing. But what's really lacking from the field and needed to convince a lot of people that inteins are in fact more than parasites in all cases is looking at the role and the importance of an intein within a native host organism. And so in the second part of my talk, I want to talk about a study that we currently have under review. This is a collaboration between the Santangelo lab at Colorado State, the Gardner lab at New England Biolabs, which actually has a basic research arm in addition to its commercial part, and my lab here at Murray State, titled Intein Exaptation Controls Archaeal DNA Replication. So inteins are abundant in these hypothermophilic archaea that live around these hydrothermal vents in the ocean, right? And these hydrothermal vents are where these minerals from the center of the earth mix with ocean water and are basically near boiling water. And this is where these hypothermophilic archaea often live, right? So one of these, a model hypothermophilic archaeon known as Thermococcus codocarensis, actually has 15 inteins, often within essential genes that are used for DNA replication, recombination, and repair. So we decided to focus on this particular protein, RAD-A. And so what we see here are three derivatives of the wild type RAD-A intein that we're actually able to make on the chromosome of Thermococcus codocarensis, right? So these are truly isogenic strains. No other changes anywhere else on the chromosome have occurred, right? And so we have a couple of variants where we have slightly decreased splicing and a little more severely decreased splicing, one where we've completely deleted the intein altogether. And so just to kind of point out, this generating these strains was really a years-long effort where we had to look at how these things behaved in vitro, do the genetics, come to an impasse, figure out how to get around it, and things like that. So these we think are really very valuable strains because they allow us to ask some really fundamental questions about inteins. Can we observe any unspliced precursor in any of these strains under different conditions? And can we observe a change in a RAD-A dependent activity that correlates with that precursor, that pre-spliced product accumulation? And then finally, can we influence the overall physiology and growth of Thermococcus codocarensis? And so what we did is we decided to grow these up and probe for RAD-A using Western blotting at 85 degrees C, which is its preferred temperature, and 65 degrees C, which it can grow at, but it grows slowly, right? And so we can see in these two intein variant strains, we have these bands appear that correlate with this unspliced precursor. And we can see in particular for this AL15 that we get about 50% precursor and 50% mature functional Rad-A at this lower temperature, right? And so if we quantify this, we can see that, over here, we're getting, maybe 40 to 50% of the functional Rad-A at this lower temperature here as well, right? Okay, so yes, we can observe precursor, particularly in this one Intein variant, this AL15. So then we wanted to look at some sort of Rad-A dependent activity to see if anything has changed, right? And so Thermococcus is unusual in the way that it likes to replicate its DNA. So it can use the traditional origin-dependent replication, which anybody who's taken a molecular biology class has learned that DNA replication starts at the origin and goes out from there. But it can also use something called recombination-dependent replication, where DNA replication is actually initiated at multiple places over the chromosome, right? And so it turns out that Codocrensis prefers to use this RDR method, and Rad-A is required for this, right? And so what are we looking at over here? So this is what's called marker frequency analysis, and it uses next generation sequencing to actually look at essentially how many reads are coming from different regions. And essentially, if we see a flat line, it's indicative of recombination dependent replication. And if we see an enrichment here in reads near the origin, that's indicative of origin dependent replication, right? Oop. Okay, and so here, when we look at the wild type situation, it looks like the previously published data, right? We get a flat line here, both at 65 degrees C and 85 degrees C. So we see that this RDR is the mechanism by which it's copying its DNA. If we look at our Delta Intein strain, we also see that this RDR is the mode of DNA replication. And this makes sense. There's plenty of functional, mature Rad-A in the cell. However, when we look at this AL15 strain, where we have this accumulation of precursors, so we have about 50% of the normal Rad-A compared to wild type, we see a clear enrichment here that gets more enriched as we go to this lower 65 degrees C, right? So this bump here tells us that these cells are using origin-dependent replication where they were not previously doing so, right? So this represents an Intein-mediated switch in the mode in which this organism is replicating its genome. Okay, so can we observe a change in Rad-A dependent activity? And again, the answer is yes. Finally, we wanted to look at just overall growth rate, right? And what we can see is in the wild type case, when we compare this to AL15, where we have about 50% splicing, we see a clear lag in growth compared to wild type, particularly at 65 degrees, but also at 85 degrees C. And while this may look subtle, if we consider the fact that this is one of 15 inteins present in Codocrensis, and many of the other inteins are within essential or really important genes, it really illustrates the power of these inteins to control the overall growth and physiology of this organism. Okay, so to summarize the second part on this new project, what we're seeing very excitingly is that the levels of active Rad-A controlled by variable splicing correlate with a switch in the mechanism of DNA replication. So a very fundamental process to the cell. And what we found really pretty exciting is this wasn't, a binary response. So in these cartoons that I showed, it's, it's off or it's on, right? But really, something like a 50% reduction in splicing has a pretty big impact on the overall physiology of this organism. And we're really excited because these results challenge over 3 decades of conventional thought of inteins as just being junk, simple parasites, and unimportant to the organism. It goes a long way in supporting all the work that we've done in vitro or in non-native intein-containing systems. And so if we consider the abundance of inteins in the microbial world and their presence in human pathogens, our findings really have widespread implications. And so, so with that, I'd really like to thank on this newer project that I spoke on with Thermococcus cotocrensis, Colorado State University, the lab of Dr. Tom Santangelo, as well as the other people who are involved in this project, the Gardner lab at New England Biolabs, and some of the work that I mentioned briefly done with my postdoctoral advisor's lab, Dr. Marlene Belfort, as well as some really talented students here at Murray State. I'll also point out, of course, none of this is possible without funding. So thank you for very generous funding from the NIGMS, as well as Kentucky IMBRE. And Kentucky IMBRE has been such a big supporter of my research. I'm very excited to say that through a recent equipment supplement, we're actually going to be able to buy the things necessary to culture Thermococcus down here at Murray State. And so we can start to look at, the effect of removing or mutating these other inteins present in the organism to try and tease out potential regulatory roles. And finally, thank you to Murray State for startup funds. With that, I'd be happy to take any questions.
Great. Chris, I have a couple of questions actually, but yeah, so just if somebody else thinking about asking you, so you guys have time to do it. So, but anyway, in Thermococcus, right? So there are, like you said, there are 15 of those genes, right? 15 inteins, 15 proteins that have.
Yep.
Yeah. Do you can you actually do you know anything about those other genes, essentially, or all other proteins, in a sense whether those inteins might be regulated similar ways as you just described for RAD-A? I mean, would it be like also, for example, a temperature effect on those inteins in the other proteins, or like if you just kind of review all those other proteins, right? So what can you say in terms of similarity and maybe differences with this RAD-A system that you just talked about? I'm just curious.
So it's a great question. So it's variables answered. So one thing I didn't share with you is that temperature actually activates RAD-A splicing in vitro is one thing that we found here. But based on literature searches, as well as some things that I'm doing in my lab right now, some of them are require temperature to splice or seem to require high temperature to splice. Others don't, and we don't really understand why. One thing that's really intriguing, this is very preliminary data, but I trust it. I'm looking at the splicing of the 2 inteins that are found within MCM. So this is an essential replicative helicase. One of these inteins, temperature is required to get splicing to happen if we express an E. coli. In the other, not really, right? So they seem to splice even at different temperatures within the same protein, which is pretty interesting. So the other things that might be affected, we know all sorts of different stresses that seem to, block protein splicing. There are different things that activate protein splicing we're starting to learn. So yeah, it's really exciting, to kind of start looking at these things in vitro and then try and understand what might be going on in vivo and how they may be acting as regulatory, providing regulatory benefit to the host organism.
And I was also curious, Chris, about, eukaryotes don't have, right, that many inteins. So it's 1%, right?
Yep.
Of those. Why?
So that's an open question. It's a very good question. So they're found within some fungi. So they're not found in multicellular eukaryotes. But it turns out that it's believed that hedgehog is actually an evolutionary derivative of an intein. And so if you look at, so the fold of an intein is called a hint domain. And let's see if we got, and it has this kind of like horseshoe, classic horseshoe fold. So hedgehog actually looks like about 2/3 of an intein and does about half the chemistry that an intein does, right? So why inteins were lost in higher eukaryotes is a good question, but we have hedgehog, right? So it maybe that was the derivative that happened.
Great, very exciting.
Thanks.
Christopher, can I ask a question? And so this is
Absolutely.
like a psychologist asking about molecular biology, so beware.
Yeah.
But is it wrong to think of these things as like long pro forms of peptides where you're cutting out different peptides from the protein and these inteins are in between, or is there any analogy there?
Well, so they're cutting themselves out of the proteins themselves. But one thing I didn't mention as well is that these things can have side reactions. So you can have this kind of productive splicing reaction where you ligate the exteins together and make the functional protein. But you can also have a cleavage event where, say, one extine will go off, or a different cleavage event where the other extine will go off. And we don't know, potentially, are some of these exteins retaining partial function or things like that. And we know that we can definitely detect these cleavage products in a test tube. And depending on conditions, we can alter the accuracy of the protein splicing reaction. So I personally think this is relevant out there in nature, but we don't have any evidence yet of that.
Great, thanks.
Yeah, no problem.
All right. Well, thank you so much. Thank you so much, Dr. Lennon. So we are moving to our second speaker, Dr. Jordan. And he's going to talk about network analysis for constructs in personality in clinical science, as you see on the screen. Great. So let's start, Dr. Jordan.
All right. Great. Thank you, Alexi. And just to be clear, can you see the full PowerPoint? You don't like see my notes or anything? Not that I have any notes, like you just see the full screen here, full title slide, stuff like that.
Yes,
Okay,
it looks great. Yeah.
Great. Just wanted to make sure, because I got dual monitors going on here. And that's when you know you made it for sure.
Right.
But really appreciate the opportunity to be part of this seminar, talk a little bit about my research in the realm of network analysis. And a couple of the studies that I've done, or at least one prominent study that I've done, that it looked at like the dark triad and how network analysis is helpful for that. And how does that apply to the discipline in which I have my expertise? So clinical psychology is what I'm trained in, and is a licensed psychologist here in Kentucky, sort of considering some future directions that would be appropriate for grant funding and bigger ideas and stuff like that, and how to envision those ideas coming together here. But psychology is a little bit different from biology. It isn't biology, it is a scientific study. And what we rely on often in psychology are constructs. So I'll mention that word a lot here. And basic definition of a construct, what we use in psychology, is just some type of descriptive label that's going to help us understand and more systematically study the things that we want to investigate. A couple of examples of constructs, especially in psychology, would be things like conscientiousness, so personality traits. And this is something that is measurable, right? Take a big 5 personality inventory. You can measure a person's level of conscientiousness. This person's conscientiousness is at the 96th percentile. So if you know about that person, it's me, by the way, when I took this test, I guess I'm at the 96th percentile of conscientiousness, you can assume some things about that person, that this individual is dutiful, organized, focused on tasks, whereas people who are low in conscientiousness are a little bit more disorganized. little bit more haptic in their day-to-day activities. Another example of a construct would be intelligence, right? And we know that intelligence has a spectrum. It's on a range, a normal curve. Majority of our population is going to sit in between an IQ of 85 to 115. But for the people who have an IQ of 130, people who have an IQ of 70, we make certain assumptions about that person's behavior, that person's intellect, cognition, based on that construct or on the levels of that construct. And one thing that's readily accessible to me as a clinical psychologist is that mental disorders are another example of a construct, right? The DSM, the Bible of psychiatry, includes a list of constructs that we use as tools for the purpose of diagnosis and assessment. Giving somebody that label, the construct of depression, is going to tell us something about the person. It's going to foster communication between individuals who understand what depression is, so on and so forth here. And it's important to formulate constructs because in doing so, we can now measure them, sort of like I was just talking about. We measure symptoms, we measure a person's IQ, we measure a person's personality, so on and so forth here. And by being able to measure them, we can now formulate theories. Knowing what depression is, knowing what a mental disorder is, we can now have a theory for why people could become depressed and why people's depression is maintained over time. But we also, like I said, we're a little bit different in psychology from the biological sciences. Like a lot of what Chris was talking about right before me, the Inteins, his field of research, those are things that are there. You can observe, you can find them, right? They exist in nature and they're waiting for us to discover them. Is that the case for something like depression? If we want to go back, is depression function or exist on the same playing field as something like an Intein, as something like these proteins? It's actually a new edition of the DSM that came out last year that included a new disorder called prolonged grief disorder. And did we suddenly discover that people experience prolonged grief responses? Did we just find it in nature, or did we sort of carve nature at her joints, so to speak? So when we evaluate good constructs, well, we need that empirical basis, right? A lot of psychometric work, we can take a good construct such as depression, knowing that depression is something that's measurable, and we assess things like its symptoms: low mood, loss of interest, sleep issues, fatigue, concentration difficulties, et cetera here. How do we assess depression? We usually do it through some self-report measure. BDI, Beck Depression Inventory, is just a common way of assessing depression. Give somebody a measure, and it basically asks, how depressed are you? And the BDI shows excellent validity because it measures what we say it measures depression, so it should be measuring depression. And it shows reliability because the items go together in tandem. People who experience sad mood likely experience loss of interest, likely experience fatigue, so on and so forth there. And a lot of empirical evidence for constructs depends on how we make up these scales, right? They have good validity, they have good reliability, so we're assuming that we are measuring depression, right? We are measuring that construct at a similar level to how we might find Inteins in the world. Typically, this is done through statistical procedures like latent variable modeling, confirmatory factor analysis. I won't go into great detail, but you can do it. It's a thing. You can assess constructs and you can determine whether or not they tap into what's known as this latent variable. We got to assume there is a latent variable. There's something to be found that exists inside the person, right? Latent variable is also known as the common cause model, and the common cause model in psychiatry and clinical psychology kind of functions like this, right? There's something maybe inside the person, we'll call that D, maybe D for disease, that's associated with insomnia, fatigue, concentration problems, psychomotor problems, so feeling sort of slowed down or agitated, weight loss, weight gain, so on and so forth. And we'll actually go ahead and call that D depression. And this is effectively what we're doing or trying to get at when we give somebody a BDI. And this is an example, first three items of the BDI. Circle 3, circle 3, circle 3. That should give me some index on what the person's depression is, right? That thing that we're trying to tap into, but we can't really see it, right? We can't hold depression. We can't find depression under the microscope, so to speak. And the common cause model works very, very well in biology, I'm assuming. Again, I like to see another psychologist on here who's just as good at microbiology as I am, and I'm not very good at it. But the disease model, the common cause model works. Let's take something called M. M causes red eyes, fever, runny nose, cough spots, cough. So if patients exhibit these symptoms, we know that they have a disease called M. Because the disease called M causes a person to have red eyes, fever, runny nose, cough spots, cough, so on and so forth. And the biologists in the room or on the Zoom call may know that the M is measles. Measles is an actual virus that exists inside of a person that you can see, that you can find, that you can test for, tangible, that causes these specific types of symptoms. And does that work for depression, right? Are we finding the depression inside the person? And is the depression causing these symptoms? You know, does depression function like a disease? I don't think so. As a clinical psychologist, right? The chemical imbalance is not really a parsimonious explanation. It's a hypothesis, not a theory. And there's a bunch of reasons why people get depressed. When you extend it to personality, does someone like people because they're extroverted, right? McCrae and Costa, formative psychologists who looked at the ocean model of personality, extroversion being included, literally say extroversion causes somebody to be a party goer. It's kind of weird to me to think about it that way. Or does somebody, like going to parties because they're extroverted? And if you go to parties, that makes you more extroverted. And the more extroverted that you are, the more you like going to parties. It's actually a seminal paper that sort of introduced the idea of network analysis in psychology, right? You can't like parties if you don't like people, right? Is extroversion just a good descriptive label for people who like other people and like going to parties? Or does extroversion cause you to like other people and cause you to like going to parties? Or if you like people, you like going to parties and vice versa. So it's sort of the idea behind network analysis that I was getting at, the nuance behind it, right? That not all, but a lot of constructs in psychology can function as what we call complex systems. The things that make up that construct, sad mood, anhedonia, fatigue for depression, party going, liking other people, being outgoing for extroversion, are all interacting with one another. It's not that depression causes this or this, extroversion causes this or this. Things that make up what we call extroversion and depression interact, and maybe depression and extroversion is just a nice little label to give these interactions here. So the construct is emergent. If you take the idea of like consciousness in biology, there's no brain area that's truly associated with consciousness. It's different manifestations of brain processes that give rise to consciousness. So consciousness is sort of an epiphenomenon of brain processes. It's sort of the idea behind network analysis for constructs in psychology. And when you extend it to clinical psychology, again, maybe symptoms co-occur because they cause one another, right? We could say, depression causes all of this stuff individually, but all these are symptoms of depression. And if you have trouble sleeping, maybe you feel fatigued. Folks on this Zoom call didn't sleep very well last night. Maybe you have difficulty concentrating. Maybe you're so focused on other things you can't fall asleep at night. Maybe that makes you feel slowed down. And maybe this causal loop, this back and forth, we're going to say, hey, depression is a nice little label to describe these processes and what's going on here, as opposed to saying that depression causes all this stuff independently. So I'll go through once I am about to introduce. I'll talk a little bit about the nomenclature. I'll refer to stuff as nodes. These are just entities, symptoms, personality characteristics, the features of that construct. Edges are going to be these connections, so lines that you'll see in a lot of graphs here. And these are things that can be statistically modeled. And centrality is a term that I'll use often here. And it's going to often refer to the node, the entity, the feature that seems to be most influential. If you kind of go back to this graph, insomnia seems to start this process, this sort of unfolding process. So it may be a central node. And the lines here are just the edges. So I'll reiterate that. When you do network analysis, it's relatively simple. I know a lot of us work with students, and a lot of students say statistics are hard, or is hard, the field. That's fine. But if you look at cross-sectional network analysis, it's really just partial correlations. You know what a correlation is? Relationship between two variables. Partial correlation is the relationship between two variables while accounting for all other variables in the data set, effectively controlling for them. In network analysis, statistical and theoretical, it's not going to assume that there's some latent or higher order factor like depression or extroversion that causes these features to come about. They're all just related to one another. That's what I was curious about, like dark triad, right? Things like psychopathy, Machiavellianism, what sort of makes that stuff up? These are all multifaceted, all related to one another. And we were really curious, are there certain personality traits that kind of explain that variation, that are sort of central or a core feature of things like psychopathy, Machiavellianism, and narcissism, stuff like that too. And based on prior literature, we focus more so on antagonism, people who are really blunt, straightforward, kind of step over other people, as a personality trait that would sort of be the driving force or factor behind what maintains things like narcissism, Machiavellianism, and psychopathy. And just to break that down really quickly, psychopathy, we're really referring to antisocial behaviors here. Folks who are disinhibited or bold, they have a lack of empathy or remorse and are particularly callous. Anybody a fan of Dexter? Dexter would be a good representation of someone who's a psychopath. I mean, he kills people, right? It's pretty antisocial. Lack of empathy, I'd assume, if you are going to kill people. Machiavellianism is somebody who's duplicitous, D for deceiving, someone who has a sort of cynical disregard, is amoral, really focuses on themselves, and is kind of schemy. Machiavellianism, I think about M for manipulativeness. And one character that comes to mind is Saul Goodman. If you've ever seen Better Call Saul or Breaking Bad, he's kind of a schemy guy, a little bit unethical, and would be a good representation of Machiavellianism. Then we got narcissism. I won't focus too much on this distinction here, but sometimes folks who are grandiose narcissists are really outwardly in their expressions, sort of entitled. The vulnerable narcissists are sort of puffed up but shaky. They have this outward expression, and one of the reasons why they do that is because they're so emotionally fragile inside. I think a good example of whoops would be someone like Miranda Priestley. If you've seen The Devil Wears Prada, she's more of a diva. I don't know what was going on the other slide, must have been a mistake. Editor-in-chief of the fashion magazine Runway, right? Very much a diva here, as our narcissists. So going back to the study that I was talking a little bit about, the Jordan et al. 2022 dark web of personality. So we looked at network analyses in 4 independent samples. I'll focus really more so on sample 1 in here. Sample 3 and sample 4 just adds a little bit more generalizability to these features. Looking at other things that are related to the dark triad, psychopathy, Machiavellianism, and narcissism, things like sadism or spitefulness, and just looking at different measures of how we assess these constructs here. Looking more so at these personality pathological traits, and those are things that can be measured here too. We look at antagonism, but there are other couple traits here that we focus on with this particular measure. And again, this is how we measure personality traits. We are asking the person on a Likert type scale, how often do I, hurt other people's feelings or crave attention, so on and so forth. It's all the stuff that goes into antagonism here. So sample 1, 294 people looking at these 5 pathological personality traits. That's the PID5BF, reform, and the short dark triad, which measures the features of the dark triad, narcissism, psychopathy, and Machiavellianism. This is what a network graph looks like. Cross-sectional, partial correlations are what these lines are. These are the edges, and these circles are the nodes. And we just label nodes here. These are the pathological personality traits, focus more so here on antagonism for the sake of time. And these are the dark triad features, narcissism, Machiavellianism, and psychopathy. So you see a lot of lines. And one cool thing about network analysis is how you can visually interpret the graph, right? You can see that a lot of the personality traits sort of hang together. Again, these edges here are just the connections, partial correlations. So the thicker the edge, the stronger that correlation is. So we see a strong relationship between psychoticism and detachment. Blue is positive, red is negative. Blue is here for colorblind folks. Usually it's green, but blue is more inclusive. So pathological traits are strongly interrelated, as are the dark triad features. But we isolate antagonism and say it has high centrality because of its multiple connections. It has connections with pathological traits, disinhibition, negative affectivity. People who are really like emotional, prone to depression, actually have higher levels of antagonism as well. But it also seems to hang with narcissism, seems to hang with psychopathy as well. Maybe as a result of that, that's what influences the Machiavellianism here. So that's what, like statistical centrality, we're really looking at how many edges there are from a node to other nodes and how strong these edges are, right? Multiple connections, pretty strong connections here as well. It's spreading activation throughout that network. If you take antagonism away, maybe some pieces fall. And that's sort of what I was saying, you know, multiple strong shared edges, that's going to be an influential feature of our network here. Sample 2, using the same measures, just different people. Slightly similar in the sample size, but we see some of the same features here too. Antagonism has a lot of connections. Narcissism, Machiavellianism in this one. A little bit weaker, but still a connection here with psychopathy. Maybe not a connection with negative affectivity, but we see a lot of spread here, right? A lot of positive associations. High levels of antagonism, high levels of disinhibition, narcissism, Machiavellianism, and psychopathy here. So that replicates, right? Same measures, same associations, just a different sample here. And you can do something called a network comparison test, just at the level of the structure, no difference overall. A couple of difference here in edges like I was talking about, but pretty much the same network. Antagonism still has a pretty strong influence here. Like I said, won't really focus much on sample 3, but we're just extending some different measures of narcissism. NVS, vulnerable narcissism, different features of narcissistic admiration and rivalry. Spitefulness is in there as well, because some people who are narcissistic are also spiteful. People who are sadists tend to be spiteful as well. You see those associations going on here, but what do we focus on? Antagonism as well, right? Centrality replicates and maybe generalizes. When you're looking at different measures of narcissism, when you're including other dark, negative personality features, antagonism seems to play a pretty strong role here. Same with sample 4, just different measures of narcissism, right? You can exploitation and entitlement, exploiting other people, feeling like you deserve good things to happen to you. Different measure of Machiavellianism, the Mach 4, which always reminded me of like a Gillette razor or something like that. Narcissistic leadership, grandiose exhibitionism, again, different measures of narcissism. Sadism here as well, psychopathy, different measures, different components of psychopathy. Antagonism still there, right? Showing these connections, different features of pathological personality traits, different features of these dark personality features as well, right? Antagonism through and through to a certain extent. So conclusions, antagonism, like I was talking about, it has some replicability here, generalizability depending on what we're trying to assess. Seems to be a really core feature for dark personality, but looking at what antagonism consists of, maybe we want to fine tune it a little bit more, see what specific features are more associated with it. It's important to note that these networks, they're cross-sectional, right? Cross-sectional, correlation does not imply causation, but it's a good hypothesis generating structure. Like, could it be antagonism, right? Can we test that in more experimental or longitudinal designs? Looking at that graph, looking at those statistics gives us an idea of how we view constructs a little bit differently, right? Traditionally, it's a psychopath who's like born evil and that's why they kill other people. Well, maybe there's other features that interrelate. It's not really that cut and dry, right? So a lot of the network analysis work, cross-sectional stuff is going to be exploratory in nature. But going back to what I'm really interested in is the networks in clinical science. And like I talked about, mental disorders are constructs. And maybe viewing them as networks can offer advances in how we treat and view people with mental illness, right? Going back to this graph. In this sense, do we treat the depression? Because if we focus on that common cause model, just give them an antidepressant. As you would a medication, an antiviral, right? For something like measles. But there's 4 different classes of antidepressants. There's more evidence-based psychotherapies for depression. If you're going to treat the depression, you may miss treating the insomnia, which you can focus on a little bit more. Maybe if you treat the insomnia, you get rid of the fatigue. Maybe for some people, certain symptoms don't matter as much, like weight loss. And what's cool is that good clinicians tend to think this way. I would never tell my patient that I'm just going to treat your depression, right? I'm going to treat your specific symptoms. You have insomnia. That's a symptom of depression. Having depression is not deterministic. Doesn't mean that your life is over and you'll always have it. But if we break this cycle, focus on some cognitive behavioral therapy for insomnia, maybe that label wouldn't fit you anymore and you'll be able to move on with your life, right? But we need to teach people a little bit more. And that's sort of the last Jordan and Weiner reference there in the Journal of Clinical Psychology. How do we like interpret these networks a little bit more? How do we bring them more to clinical science? And what's really cool research, and really what I want to move forward with, is that patients' preliminary data suggests that they understand their problems being conceptualized as this way. And it's less deterministic, right? As opposed to having depression, you can do something with it. You can give this conceptualization and patients can buy into treating or to being in therapy or whatever mode of treatment. But we need a little bit more work with the statistics piece behind it. But really my idea here, when we think about mental disorders, like embrace the complexity, right? Don't just say it's depression and that's it. Depression means a lot of different things, but what constitutes depression? And some, really interesting research is having patients sort of construct their own networks. And you can do that through like EMA, ecological momentary assessment. They get pings to like their cell phone. And, we sort of rate, how depressed do you feel in this moment? 2 hours later, how depressed do you feel? You can sort of get them to conceptualize their own networks and see over time, like I'm caught in this feedback loop where if I feel depressed at 12, I'm likely to feel depressed at 2, 4 p.m., 8 p.m., vice versa. So how do I break that mold, right? How do I intervene somewhere to where this cycle stops? And there's not a lot of research that's focused on that, but Murray State has a pretty good psychology clinic, has some pretty good clinicians in there, has some pretty good supervisors, right? And I would love to see the idea of network analysis being more formally rolled out. It's a grant seminar, and that would be something that I'm looking forward to when I think about funding and aspects like that. So I want to thank all of you who are on the call, the opportunity for this, talk a little bit about my research and folks who have helped out with these projects as well. So that's all I got from my end, and I'm happy to take any questions.
Great. Yeah, any questions?
Yeah, I had one. So this antagonism, in all these different types of, I guess, this Machiavellianism or psychopathy, things like this, do you see that? I'm wondering if it's outward or if it's more hiding, right? Like, if that, it may be kind of a naive question, but do certain types of personality traits, are they more likely to be, antagonistic on the inside, but not necessarily on the outside? Or is that inherently kind of an outward trait?
Yeah, it's a great question, and kind of like what I was saying, the need to sort of fine tune the construct itself even a little bit more, because this is the PID-5, and these are the items that are highlighted that measure antagonism. Like we did, well, not I, but people who've developed this scale did all those like confirmatory factor analyses, and they're saying that, I crave attention is something that's supposed to be indicative of an antagonistic person, which you wouldn't think on the surface, right? Like somebody who's antagonistic is not like necessarily putting other people down or fighting with people just for attention seeking, right? It's a trade-off between, the PID-5 BF only has 25 items. The full measure has like 300 items. And, how does that go into analysis? How do you get participants to take those longer measures? So kind of a trade-off. Definitely need to focus on the construct too a little bit more.
Cool.
So Gage, is the idea that if you identify this factor called antagonism, that you would then treat that like you would treat concentration problems or insomnia? Is that the idea here?
You could. I mean, usually this for personality science, if that's the focus, and you can certainly work with people who are antagonistic, like even a psychopath, not everybody who has that label is like Hannibal Lecter or anything like that. People understand like the consequences of their actions. And if you want to sort of translate this to treatment, then yeah, I think it would make sense if you focus on antagonism. Maybe you'll see less of those features of other, dark triad traits and stuff like that.
Gage. And also, like, I have a question on, so when you say treat patients, so I guess I got from your talk that, one of these ideas that it's actually helpful, right? You just tell me if I'm wrong, but it's helpful for patients to actually see this network, right? So essentially you kind of make it individual for like a particular patient, and then it's kind of beneficial for that patient to see that specific network for that particular person. Is this something that I got right?
Yeah, absolutely. And what I'm all about doing, because I supervise, I teach folks how to do therapy for the first time. And a lot of what I talk about is how do you sell the therapy to the client? Because therapy is no walk in the park. It's not easy. It's fun sometimes, but not always. And if you break it down like this, as opposed to saying, let's just treat your depression, let's go along with it, because this is what works, maybe you get a little bit more buy-in. You are depressed, you have that label, but what happens if we focus on insomnia? Maybe you won't feel as bad, you won't have that label anymore. And it shows that you've given attention to the patient, right? They're not just coming in, you're saying, I just know how to treat depression, so I'm going to do it like this. They have a say in their conceptualization, their treatment as well, and that's going to really matter in the long run.
Great. Well, any other questions? Well, all right. Well, I would like to thank you very much, Dr. Lennon, Dr. Jordan. So wonderful presentations. So yeah, so with that, I'm gonna close the seminar, and thanks again.
Thank you.
Yeah, thank you.
Thanks, Whitney.